So we continue to talk about the different aspects of microbial growth today. And next topic of discussion is how we actually assess the growth, right? And to do so, obviously, we need to count cells, right? So we can count cells using microscope directly, okay? And if you remember the samples that you've taken, samples the wet mounts and the fixed specimens, there were a lot of cells there. And when you look in the microscope, there's a certain field of view, and you don't really know how big that field of view. So if you counted 5 cells in what? In 1 microliter, in 2 microliters, in 50 microliters. To do this in the quantitative way, we count cells in so-called Petrov-Hauser chamber. So it's essentially a slide which has a metal grid on it. You apply your sample onto that grid, and cells randomly distribute across the grid. Okay? And then you look in the microscope and you look at quadrants on the grid, and you count number of bacteria in each quadrant. Different techniques for different types of, you can try different quadrants. The smaller the cells are, the smaller the quadrants you can address. But say you count number of cells in 5 quadrants, and then you multiply it by a certain factor, and you get the number of bacteria per microliter, for instance. It's really simple, okay? So all you have to do is to take bacterial specimen, add some stain, throw it on the chamber. Sometimes it is called, well, it's similar to hemosideometer. And then you count number of cells in the squares. Okay? Does that make sense? The problem with that method is that you cannot easily distinguish live and dead cells. Okay? There are stains that would stain only live cells, okay? And then you can do that. But using crystal violet, you wouldn't be able to distinguish dead and live cells. Does that make sense? Good? Right. Now, the advantages of the technique, it's simple in terms of the equipment. All you need to have is a microscope and the slide. Disadvantage, if you deal with a lot of samples, it takes a lot of time and a lot of effort. I counted not the bacterial cells, but mammalian cells using that technique. And it's boring. It requires a lot of concentration. You really have to be all in the picture, just counting cells. And it takes time. So if you have, 100, 150 cells in all 5 quadrants, it's going to take a lot of time counting. Okay? And of course, if you have to distinguish between something that looks like cell and something that looks like debris, deciding whether it's a cell or not, it is your decision. So there is some kind of a bias. Okay? If you would take the same sample, exactly the same sample, and ask 2 different people to count number of cells, you will get different results. They're not going to be horribly different. We're not talking about 100 versus 500. But one person may have 100 cells, another person may have, 110. Does that make sense? In order to avoid that bias, in order to automate cell counting, cell Coulter counter was developed. So Coulter counter uses quite an elegant principle for counting cells. Cell, every time it passes between two, passes through this certain aperture in that chamber, okay, and cell goes through, the electrical resistance of that electrical chain, okay, changes. Does that make sense? Cell passes through the aperture, changing the electrical resistance here. Every time cell passes, electrical resistance changed. It can be registered by the automated system. So every cell will be counted automatically and pretty quickly. So that's the advantage of the Coulter counter. Rapid, no manual work, no bias, can be automated. The disadvantages: first, it cannot distinguish between live and dead cells. You understand why? They change the resistance the same way. And second, it only works for diluted cell suspensions. Why? Why you cannot count cells in the cell suspensions that are really dense? When cell suspension is dense, there's a higher chance of what event? Each time one cell passes the aperture, it is registered. If you will increase number of cells, what can happen? Multiple cells will go through the aperture at the same time. That makes sense? So that's the downside of the Coulter counter. Of course, the gold standard is the viable cell count. And the ideal method is to grow them on the plate. Assume that we have a liquid culture, liquid culture that has a lot of cells, and that's pretty much the pour plate that you did last week, okay? And you prepare serial dilutions, decreasing the number of cells with each dilution, okay? And then you can plate this dilution either using the pour plate method, right? Or using the spread plate method. Spread plate, as we discussed, is a fairly simple approach. You take certain volume from the dilution, place it on the Petri dish, and then spread it over the surface with a hockey stick. Spread plate method is more reliable because in a pour plate, remember, we had pretty different results between different groups. Maybe due to the handling of the pipette, maybe due to the death of the cells when they are exposed to the hotter agar. So that creates more bias than spread plate method. That makes sense. Now these two methods are convenient when you deal with highly concentrated samples, like liquid culture of bacteria, the ones that are there. There's a lot of cells there. We figured that even, we had results estimating the number of bacterial cells in 1 milliliter in the ballpark of 1 billion. What if you deal with extremely diluted sample, like water? drinking water. If you want to check if there's some bacteria in drinking water, or you want to go down to breakers, grab some of the greens, the salad greens, and see if there are any bacteria from human feces on it. Okay. There's not going to be a lot. There are not going to be millions and millions of bacteria, but there are going to be few if you test, I don't know, drinking water or well water. There might be some. So how do you deal with extremely diluted samples? In this case, you use filtration. Does that make sense? So say you have 2 liters of water from Lake Erie. You run it through the filter, and all bacteria that are in that sample, they remain in the filter. And then you can deal with it many different ways. You can actually do the replica plating on the agar and see what's growing on. Okay? You can directly apply some special differential media that will highlight the bacteria, say coliform bacteria or lactose fermenting bacteria, and so on and so forth. Okay? But the idea is that you concentrate the bugs from the diluted sample using filtration. Does that make sense? Now, 2 last methods, turbidimetry and the most probable number. If you don't mind, I'm going to start with the simpler one, the turbidimetry, and then we move on to the most probable numbers. So turbidimetry is illustrated in the left bottom corner of the slide. Samples. You see the upper sample, this one, is transparent. The bottom one is cloudy. Cloudy, another word for it is turbid. So you measure how much light is absorbed by each of the samples. Does that make sense? The more cells you have, more light is absorbed. Is that good? Is that understood? Obviously, at the certain moment, the certain cell concentration, number of cells may increase more and more and more, but there will be practically no change to the density, optical density. Does that make sense? So the ability of the solution to absorb light is called optical density. Okay? And we essentially, in turbidimetry, we measure either, well, optical density, or you can call it light absorption. Okay? Now, the advantage of this method, it's really fast. If you have spectrophotometer at hand, you can just sample the culture, throw it in the cuvette, throw it in the spectrophotometer. It's really fast. We talk about, I don't know, 5 minutes. And you get the number. So you can make a quantitative growth curve using turbidimetry. The downside, you won't be able to tell apart live and dead cells, because all of them can absorb light. Is that clear? Most probable number. Should I start? So this method is used to estimate the number of cells mostly in the environmental samples. Samples that are usually diluted. Again, imagine that we are talking about the water from Lake Erie. You know, we took sample at Headlands Beach. Okay? So we have pretty considerable amount of water, say a gallon. This gallon contains maybe tens, maybe hundreds of the bacterial cells. Does that make sense? Okay. So what do we do next? We prepare 3 sets of 5, so 15 tubes with the indicator media. When we say indicator media, it means that when we will be able to see the bacterial growth, okay, for the bacteria of interest. Say we want to take a look at the coliforms, okay, E. coli and its close relatives. That makes sense? So then we do the following. We take 5 samples of 10 milliliter from that lake water and inoculate the set of first 5 tubes. Take 1 milliliter of the lake water, inoculate the second set. Finally, take 0.1 milliliter and inoculate the third set. Do you follow me? Now, which inoculum, which volume will likely contain more bacteria? 10 milliliters, 1, or 0.1? 10. Is there a good chance that every 10 milliliter inoculum, every of those 5, will contain some bacteria? Yes. 1 milliliter, the chance is less. Does that make sense? And in 0.1, you may or may not have microbes in each of the samples. Does that make sense? So the result will be that in the 10 milliliter inoculum series, all tubes show growth, 5 positives. In 1 milliliter series, only 2 tubes show growth. And in 0.1 milliliter series, none of them showed growth. So what does that mean? Let's say for the 0.1 inoculum, what does that mean? That none of those samples contained what? Bacteria, yeah, coliforms, whatever we're looking for, right? So it was so small that nothing was in the sample. Does that make sense? Here, in the 1 milliliter inoculum series, only 2 samples contained bacteria and 3 did not. Does that make sense? What do we do next? Next, we open the special book, the reference table. And the reference table has numbers. Okay? We have number 5, right? 5 positives in 10 milliliter inoculum. 2 for 1 milliliter inoculum, and 0 for 0.1 milliliter inoculum. Do you follow me so far? The answer that is called most probable number is 49. 49 cells per 100 milliliter of lake water. How should we interpret this result? Especially knowing that there are confidence limits. When I say how should we interpret, I ask you, what does that mean? Can you explain it? What does that mean? That we determine that it's 49 cells per 100 milliliter is the most probable number. Strictly 49. So having 49 has the highest probability. Does that make sense? We take any 100 milliliters of that water, that sample. There's a very high chance, the highest chance, to have 49 cells in this 100 milliliters. for a given 100 mils, can we have 25? Yes. Can we have 90? Yes. If we compare probability of having 90 to having 49, what is more probable? Having 49. That's why we call it most probable number. Does that make sense? So if we will try to plot the probability, it's going to be this. So this is 49, and those are other numbers. Does that make sense? We still may have them. Now, why it's convenient? We can correlate the risk of, I don't know, acquiring the digestive infection while swimming in the lake water with the most probable number of microbes that we detect on a certain day. And we can figure out the limits. Say, we set the limits if the number of microbes in the lake water is, I don't know, 49, is most probable number is 49 or more, then we close the beach. If it's less, then we're fine. Does that make sense? So this method it doesn't give you the exact number, undisputable number. It gives you a probability to have certain number of bacteria in the water, okay? Does that make sense? Now, let's see what microbes need for growth. They need nutrients. And we're going to quickly discuss where they take them, what they're made of actually, the microbes. Most of it, of course, is water. Okay? You can see that typical E. coli is 70% water. Proteins, nucleic acids, carbs, lipids. So you can see that proteins comprise 15% the weight of an E. coli. Kind of shows us that life is made of proteins. That makes sense. Now, proteins, carbohydrates, lipids. What is the main chemical element that is common for all of them? They're all organic molecules, which means they all have carbon. Okay. So carbon in those biochemical molecules comes from inorganic sources, in case of autotrophs, which can be carbon dioxide in the air or bicarbonate ion in the sediments, or it can come from organic sources for heterotrophs like us. Okay. Oxygen, another critical element that is present in all organic molecules that microbes have inside of them. Okay, practically all of them. Oxygen comes from the air, obviously. Nitrogen is extremely important. And nitrogen is actually a very interesting element which is extremely inert. What is the percentage of nitrogen in the air? 78. 78 and 79. A lot, okay? Most of what we breathe in is nitrogen. And it's chemically inert. What does that mean, inert? Yeah, inactive. It doesn't get into the reactions normally. So how inert nitrogen is? If we would take, I don't know, a pile of firewood, okay, and light it up, it's going to be carbon in the firewood. It gets oxidized, gets burned by the oxygen in the air. Nitrogen doesn't do anything. It doesn't get into any type of reaction when we burn the firewood. It's pretty hot, but this energy is just not enough to do something about nitrogen. So, but nitrogen is extremely important. In which biological molecules can we find it? Macromolecules, big molecules. We have not a lot of them, so it's kind of 60% success rate, even if you just shoot blindly. Where can we find nitrogen? Huh? DNA, of course, nitrogenous bases. So DNA, RNA, nucleic acids. Where else? Proteins, yeah, amino acids. That's why they amino acids, right? Lipids, not really, no, unless they're modified. Okay. Does that make sense? So proteins and amino acids. This is extremely important to get nitrogen in there, but it's inert. Somehow bacteria manage to what we call fix nitrogen. They do amazing stuff. Bacteria of the genus Rhizobium that are symbionts growing on the roots of certain plants, mostly legumes, they perform this conversion. They convert nitrogen into ammonia. Okay? Does anyone do vegetable gardening? Anyone ever grew like soy or beans? Well, we did. Which conditions does soy grow at? We need like something extreme, like 100 atmosphere pressure, 200 degrees Celsius. What condition does the soy grow at? Come on, people. Like, yeah, room temperature, normal atmospheric pressure, you throw the beans into the soil and they will start to grow, right? So humans can do this reaction too, industrially. Conditions are slightly different. It's called Haber-Bosch, don't write it down, Haber-Bosch process. So conditions for Haber-Bosch process that in the plants, done in plants, 5 to 600 atmosphere pressure, and temperature is about 5 to 600 Celsius. And you also need a platinum catalyst for it. Plants can do it here and now, at this normal temperature. How? I have no idea. If we would, it would save trillions of dollars probably. Okay? So this ammonia is the initial molecule, it's essential molecule for synthesis of amino acids and nitrogenous bases. Does that make sense? There are other microbes that will oxidize this ammonia, producing nitrous acid and then, nitrous acid. Nitrous oxide. I think it's called nitrous oxide. And then nitrous oxide can be reduced back to nitrogen. What I briefly illustrated up here is called the nitrogen cycle. Nitrogen is reduced to ammonia, ammonia is oxidized to nitrous oxide, and then nitrous oxide is reduced back to nitrogen. Does that make sense? That's critical. Microbes are extremely important. They actually play a key role in cycling nitrogen in the environment. Does that make sense? Now, I'm going to stop with nutrients at this point. I want to go back for a second and tell you the take-home message for the cell count. So what you have to know about the methods. I am not so much focused on the description of the methods, but for each method know whether it can distinguish live and dead cells. Okay? And second, what are the advantages and disadvantages? So if I tell you that turbidimetry is extremely prone to operator bias, is that true or false? turbidimetry, when you measure optical density, you measure it in the machine. So you stick something in the machine, it gives you a number. Is there any bias from you? No. When you count cells, is there a bias from you? Yes. When you count colonies, is there a bias from you? Yes. If you are involved in quantitative estimation, there's a bias. If it's a machine that does it for you, there's no bias. Does that make sense? Like the thermometer reading. When you take your temperature, you may feel like you're fine, but if the thermometer says 104, that's 104. You cannot argue. Does that make sense? Any questions? Not really? Okay. Let's wrap it up.